The overall goal of this technique is to systematically investigate the genome-wide DNA methylation landscape in large patient cohorts.
This method can help answer key questions in the epigenetic field, especially with regards to which regions in the DNA methylation landscape get deregulated in patients.
The main advantage of this technique is large number of the patients can be investigated in a cost-effective manner.
This technology can have positive implications to the therapy and diagnosis of the patient because we can investigate existing biomarkers and also identify the possible and novel biomarkers.
Though this method can provide insight into patient cohort, it can also be applied to other systems, such as DNA methylation in cells derived from cultured cell lines or from mouse rat samples.
Demonstrating this procedure along with Dr. Ya-Ting Su will be Mr. Joseph Liu, who is a technician in our lab. The sequencing part will be demonstrated by Ms. Garcia, who is a technician in our sequencing core.
After isolating genomic DNA from tumor or normal tissue according to the text protocol, add 1.2 micrograms of total genomic DNA to 96 microliters of TE buffer and 24 microliters of 5X bind wash buffer to make a 120 microliter solution. Sonicate the DNA solution for 30 seconds, then allow it to rest for 30 seconds. Repeat the sonication a total of 20 to 25 times. Use a bioanalyzer system with a commercial